The general research is about axonal regeneration and mechanisms underlying the phenomenon. It is a really important topic with all neurodegenerative diseases and other accident consequences leading to paralysis. The subject is really complex. The aim is not only to regenerate axons, but also to regenerate fibers on long distances. This aim of the study is to find one of the regulators that can act on axonal regeneration. Neurons are studied after axotomy, which faces two critical decisions: survival or death. For a surviving neuron to regenerate its injured axon, a required step is the assembly of a growth cone at the leading edge. A growth cone appears when a damage occurs on an axon. It is a structure which exists in axons in development. Based on the correlation between growth cone reformation and successful axon regeneration, it has been proposed that growth cone formation might be a limiting process for initiating axon regeneration. For example, following a crush injury to peripheral nerves in adult rodents, many axons assemble a new growth cone within hours. However, in adult CNS, injured axon terminals often appears as dystrophic end bulbs or retrogression bulbs, possibly representing abortive attempts of growth cone formation. The Doublecortin family, also named DCX, has been implicated in regulating neuronal migration and axon growth during development. Previous studies have suggested that, similar to DCX, DCLK1 and DCLK2 regulate neuronal migration and axon growth during development, and overexpression of DCX or DCLK enhances neural growth in vitro. To assess whether forced DCLK2 overexpression might alter the injury response of AGC, AAV2 vectors expressing DCLK2 or a control encoding for PLAP are injected into the vitreous body of wild type. An optic nerve crush is performed on these mice and analyzed both RGC survival by immunostaining. In contrast to controls, in which few injured axons sprouted across the injury sites, DCLK2 overexpressions resulted in a modest yet significant increase in axon regeneration. Approximately 200 grew 500 mm beyond the lesion site, consistent with the notion that downregulation of DCLK2 might contribute to regenerative failure after optic nerve injury. Interestingly, they found that DCLK2 overexpression led to a significant protective effect on RGC survival. In contrast to 20% survival in the control group, about 50% of the RGC survived after DCLK2 overexpression. Moreover, phosphoryl C gene is known as an injury signal, which is important as a trigger of neuronal death. They examined whether DCLK2 expression might affect injury-induced expression of phosphoryl C gene in RGCs. In wild-type mice, an optic nerve crush rapidly induces the appearance of phosphoryl C gene in the cell body of most RGCs. However, DCLK2 overexpression significantly reduced such injury-induced phosphoryl C gene in RGCs at both 24-hour and 72 hours post-injury, suggesting that DCLK2 expression may protect injured RGCs by inhibiting the generation and/or propagations of injury-induced signal. Previous research has shown that the deletion of P10 increases the axon regeneration. With this result and DCLK2 results, an idea came to combine both action of P10 deletion with AAV2 DCLK2. P10 is present on mice, but is framed with locks. An injection of AAV3 deletes the gene. 2 weeks after injection, results showed that DCLK2 overexpression further enhanced neuronal survival from approximately 50% to 70% when compared to the P10 deletion alone. The results presented here implicate the DCX family members localized in the axonal compartment as critical regulators of neuronal injury responses, with strikingly diverse effects on neuronal survival, growth cone formation, and perhaps axon extension. In addition to promoting the initiation of axon regeneration, DCX members also greatly enhance axon elongation induced by P10 deletion, suggesting a new set of targets for promoting axon regeneration after injury. Together, these results suggest that this axonally localized protein might act as a key sensory hub in coordinating different aspects of neural injury responses and could be a set of important targets for potential intervention toward neuronal protection and repair under diseased condition.